Judges look like they're done writing. And so it's now time for me to introduce our 9th speaker. Our next one is Katie Cabral. Katie is in the bioengineering PhD program. Her dissertation advisor is Zev Gardner, and her talk today is entitled Building Blood Vessels.
The United States has a serious shortage of organs available for transplant. 20 people will die every single day while waiting for their organ. So how do we solve this shortage? Bioengineers like myself are interested in making new human organs from scratch. And that's a pretty crazy idea, right? That you can take some cells and put them together in a way that gives you a new liver or a kidney. And although there are many challenges that still need to be overcome before this idea can be a reality, today I'm just going to talk about one. How do we put blood vessels inside engineered organs? Cells need blood in order to provide our cells with oxygen and nutrients. And if we want our engineered organs to survive upon transplantation, we need to fill them with a blood vessel network that can provide their cells with blood. And so scientists have used techniques like 3D printing to try to artificially create blood vessels. And we can make blood vessel-shaped structures out of the cells that make up blood vessels, which are called endothelial cells. But just having the right cells and the right structure is not enough to get these cells to form functional blood vessels. And so something is missing, and we hypothesize that something else is needed in order to get these cells to form blood vessels. Something that tells them it's that time to organize themselves. And so the goal of my graduate research is to understand how the environment affects the behavior of endothelial cells. And the idea is that if we could give these cells the right environment, we could guide them to form better blood vessels inside engineered organs. And so one of the experiments that I have done is to look at how the density of endothelial cells affects the formation of capillaries. Capillaries are your smallest blood vessels. Think a skinny tube, just 1 to 2 cells wide. And what I did was I printed endothelial cells into a line the size of a capillary, but the cells didn't form a capillary. They acted like individual cells and they started moving away from each other. But when I took that same capillary-sized line of cells and I added in a lot more endothelial cells into the surrounding environment, this was enough to make each of those cells in that line join up with its neighbor and form a continuous capillary. So what's happening is that the cells all seem to be communicating with each other and deciding as a group whether they should all form capillaries or to remain mostly as individual cells. And the more cells that are present in the environment that can contribute to this conversation, the more likely it is that these cells will choose to form capillaries. And so my next step is to figure out exactly how these cells are talking to each other so that when we try to build blood vessels, we can give our cells those same messages. And figuring out how to build blood vessels will make it possible for us to replace damaged and diseased organs with artificial new ones. Thank you.